aging is the major risk for all of the major diseases that we're facing. If we really are able to target something fundamental to the aging process rather than going after these diseases one by one, we really might be able to have a much greater impact.
Professor Joseph Baur, professor of physiology, expert in metabolism and aging.
We've been able to delay aging in rodents by calorie restriction. So simply maintaining their nutrition, but reducing the number of calories they're able to take in. And that's sufficient to extend lifespan by 30 or 40%, leave them looking healthier and younger much longer. And so we're really making a push, I would say, as a community these days to try to apply this logic to humans, or at least to figure out if something like this is possible in humans. Very little evidence for benefit from supplementing antioxidants in humans, even though there's been an enormous amount of effort put into that. As you get older, in some tissues, the level of NAD goes down. Does that matter? This thing that is so fundamental to your metabolism is actually decreasing. If we restore it, will that make things better or not?
Hey there, welcome to the Happy Habit Podcast. I'm Matthew. You'll find me here Mondays and Thursdays talking to many of the world's most interesting experts on the subjects of health, well-being and self-improvement. So if that is your thing, well then feel free to dip into the Happy Habit archive. I've recorded over 400 episodes to date, so bound to be something of interest to everyone. And if you're getting value from tuning in, well then show your support. You can like, subscribe, share with other people who have similar interests. And if you're on YouTube, become a subscriber over there too, as many of you have done to date. We all want to live as long as is possible, and for many years, scientists have been exploring the subject of longevity to unlock its secrets with varying degrees of success to date. And so to explore aging and metabolism, I'm joined today by physiologist Professor Joseph Baur. We ask what causes aging and why there seems to be no consensus on this very vital question. We hear about calorie restriction and how it has been shown to increase the lifespan of animals by up to 40%. Dr. Baur busts the myth of antioxidants' ability to stave off aging, which was a surprise to me. Expect to hear about the molecule NAD, how it declines as we get older, which seems to have an influence on the aging process. We also discover NAD's relationship with an important enzyme called CD34. Dr. Baur also tells us of the groundbreaking work he and his team have done to discover an important NAD transporter that carries NAD from the cell cytoplasm into the mitochondria. We also ask, how do we boost NAD levels and what the significance of NMN is? Plus, we hear about the market for anti-aging NAD supplementation and why one needs to take anti-aging assertions made by companies with a pinch of salt given the lack of clinical trials to date. This is a really interesting discussion on longevity. I hope you enjoy. Really what I want to do is I want to introduce to people the subject of aging research, because it's obviously something that we all have a vested interest in. So in a general sense, just to find out what aging is and what you do, then just to flesh out the picture for people, if that's okay.
I mean, I guess, the broader picture of aging research these days is that people are moving towards what we've termed the geroscience hypothesis, which is the sort of general concept that aging is the major risk for all of the major diseases that we're facing. And that if we really are able to target something fundamental to the aging process rather than going after these diseases one by one, we really might be able to have a much greater impact. And that's something that sort of may sound like a pipe dream at first, that maybe we can reverse aging to, combat cancer or cardiovascular disease, but it's something that's very much a reality in laboratory animals. So we've been able to delay aging in rodents for years by particularly by calorie restriction. So simply maintaining their nutrition, but reducing the number of calories they're able to take in. And that's sufficient to, to extend lifespan by 30 or 40%, to delay all the different age-related diseases that we routinely monitor in the animals, and to really leave them looking healthier and younger much longer. And so we're really making a push, I would say, as a community these days to try to apply this logic to humans, or at least to figure out if something like this is possible in humans.
And speaking of that community, is it true that we still don't really have a consensus as to what the underlying cause of aging is?
Yeah, absolutely. And it's funny, there almost was a consensus for a while, right? There was this idea of the free radical theory of aging, that we were producing reactive molecules as part of our metabolism, and that those would damage the macromolecules, our DNA and proteins, and that over time that accumulating damage would overwhelm us. And so this is where the idea of taking antioxidants came from. And this is a really attractive hypothesis. It makes total sense molecularly, and there's something to do about it, by taking antioxidants or overexpressing antioxidant enzymes. And this theory just hasn't held up in the simple form. At least all the different mouse models that have been made to test this, or at least one or two have marginally supported it. But the vast majority have refuted this idea that if we control free radical levels, that aging will be mitigated. And certainly there's very little evidence for benefit from supplementing antioxidants in humans, even though there's been an enormous amount of effort put into that. And so unfortunately what's happened is I think most people have come to accept that at least the simple form of that theory is not right, but not much has replaced it. And it's actually kind of amazing, how much we've managed to go on without putting a molecular mechanism in place. I think there's a few interesting ideas, but as you said, no consensus. I mean, there's certainly people that are going to argue with any one that you put forward as what you think is the key mechanism right now.
In the landscape of aging research, different scientists in different part of the world are studying different molecules that have significance in aging. Now, you study in particular a molecule called NAD. Could you tell us what NAD is and what role it plays in the body, and then how it is significant as far as aging is concerned?
Right. So we hope it's going to be significant as far as aging is concerned, but I can tell you why we're interested in it. So it's sort of when you get into the weeds of metabolism, of how we break down the molecules that are in food to produce usable energy for the cell. There's a whole series of biochemical transformations going on. And if you look at your biochemistry textbook, NAD is one of these things that's coming in and out at different steps as you go through different enzymes. But it actually accepts the high energy electrons from food. So as you're pulling the energy out, NAD is essentially the shuttle mechanism that captures some of that energy and brings it to the mitochondria, where it can be used to produce ATP, which is what your cell uses as energy. And so there's about 500 reactions already known and described that individually require this molecule. But I think the most fundamental aspect is that you can't extract energy without it. And so it's completely fundamental to life. There's a disease called pellagra that was endemic in the southern U.S. and in many populations throughout history where people have been malnourished. That is the result of not getting enough precursors for NAD in your diet. And so we know that's lethal. We know the symptoms of being very low on NAD, and that it eventually becomes fatal. Even at the cellular level, cells can't live without NAD. But the question has kind of become, with aging, do the changes that we see during normal physiology in NAD levels actually matter? And that's what we're starting to realize, is that at least that there are changes as you get older. In some tissues, the level of NAD goes down. It's not the whole body and not to the degree that causes this lethal disease. But now we've got this real question of, does that matter? Right? This thing that is so fundamental to your metabolism is actually decreasing. And if we restore it, will that make things better or not? Aging is sort of a more generalized, small, mild decrease, but we certainly see in things like infectious diseases, there are cases where the NAD level can really crash in certain tissues.
Well, we know that NAD depletes at varying degrees, not uniformly across the body, for example, but do we know how it's depleted as we age?
So there are a lot of opinions on that subject as well. So there are various NAD-consuming enzymes that go up with age. And in addition, many of the enzymes that are involved in biosynthesis of NAD go down with age. And so we've done some work using heavy isotope tracers, and our work supports the idea that it is the consumption of NAD that's increasing with age, that our synthesis capacity, or at least our synthesis rate, remains relatively constant. And when tissue levels are declining, that to us looks like it's because you're increasing the activity of some of these enzymes that chew it up. Our work is sort of agnostic to which enzyme that is. And I would say there's two sort of leading candidates. Probably the one in most people's minds as the real culprit is probably CD38, which is an enzyme that's expressed on immune cells and endothelial cells that chews up NAD and also chews up the precursor nicotinamide mononucleotide. And so it seems like animals that have that enzyme deleted maintain their NAD levels much better with age. But there's also an increase in PARP1, which is a response to DNA damage. And that's an enzyme that chews up NAD to make modifications on other proteins and factors that are involved in the DNA damage response. And if you get enough DNA damage and enough PARP activation, you can actually wipe all the NAD out in the cell as well. So I think both of those are still viable candidates to be contributing to age-related NAD decline under different circumstances. And there's many other NAD-consuming enzymes that we haven't really I think in many cases evaluated well enough to be sure.
And those enzymes, were they discovered through your artificially depleting NAD in mice, or was that something different?
That was something different. For the most part, it hasn't really been our work uncovering the role of these enzymes. Perhaps have been known about for a long time, and there's a couple of different groups that have really focused on CD38, particularly Eduardo Cini's group at Mayo, and Eric Verdin's group has gotten into that mix a little bit. But we've only been peripherally involved in actually characterizing the activity of those enzymes.
Is it true that you discovered the transporter that facilitates NAD getting into the mitochondria, the powerhouse of the cell?
Yeah, so that was one of the bigger things to come out of our lab in recent years. And this was something that we, as we manipulated NAD levels up and down, we found that the phenotypes we were seeing were often related to mitochondrial function. And so this idea that maybe it was the mitochondrial NAD pool that really mattered. And we're really kind of shocked to find out that it wasn't known how NAD gets into the mitochondria. And so we spent a little bit of time doing heavy isotope tracing studies where we were able to sort of put labels on the NAD that's in the cytosol and show that same NAD shows up in the mitochondria, which made us realize there was a transport mechanism of some kind. And so we went looking for it and found this protein that had been designated SLC25A51, which was already known to be localized in the mitochondria. It was known to be in a family of proteins that are transporters of metabolites. And it had shown up multiple times in screens for things that were lethal when you deleted them. So we knew it was an essential gene, and it was sitting in the right spot, and it looked like the right type of protein. And so just through a candidate approach, we were able to show that does seem to be the critical one for getting NAD into mitochondria.
I spoke with a cancer doctor recently who was very passionate in his belief that cancer was a metabolic disease rather than a disease of genetics or genetic dysfunction. What I'm interested to find out is, what are the impacts of NAD on cancer cell growth?
Yeah, so particularly in relation to the transporter, I think there may be a pretty interesting answer to that. And one of the few things we could find when we went looking through public databases for information about this protein after we had identified it was that in the Human Genome Atlas, there's an evaluation of different genes as prognostic markers for cancer. And it turned out that in liver cancer or thyroid cancer, which are generally pretty active in terms of mitochondrial function, it's a bad thing for you if your tumor has high levels of SLC25A51. But in kidney cancer, which is usually more glycolytic and less reliant on mitochondrial function, it was a good thing to have high levels of this transporter. And so we've got this idea that maybe you can match or mismatch the type of metabolism your tumor likes to have, based on whether you're pushing the NAD into the mitochondria for the ones that like that, or if you take something like kidney cancer where it wants to be glycolytic and use the cytosolic NAD, if you push it all into the mitochondria, you might actually thwart the tumor growth.
I suppose the big question at this stage for the audience is, are we able to affect our own levels of NAD through our behaviors, be it through calorie intake or indeed exercise?
Yeah, so there's good evidence that we can increase muscle NAD levels through exercise for sure, and pretty decent evidence that if you do at least a calorie restriction in animals, you can get an increase in NAD in muscle and also in adipose tissue and fat. So I think, yeah, for sure we can modulate them quite a bit. And then if you look at this disease pellagra, right, this is from a dietary deficiency of all the NAD precursors. So there's certainly a point where you need to get a sufficient amount of these precursors, which are vitamin B3, essentially, nicotinamide or nicotinic acid, which are vitamin B3, or tryptophan and protein, which your body can process to make NAD as well. And so there's certainly, it's important to maintain an adequate intake of some combination of those to keep NAD levels from dropping into that dangerous range where you can get these disease phenotypes.
You mentioned it briefly earlier. Can we talk about NMN? Could you tell us exactly what it is and what relationship it has to NAD, if any, and then how it is significant from an anti-aging standpoint?
So NMN is the immediate intracellular precursor to NAD. So it's nicotinamide mononucleotide. So vitamin B3, I mentioned, is sort of the typical precursor that has been given to boost NAD levels over the years, which is nicotinamide. The simplest form to start thinking about nicotinamide or nicotinic acid. They run through parallel pathways. But if you start from nicotinamide, you go through an enzyme called nicotinamide phosphoryltransferase, NAMPT, that converts the nicotinamide to nicotinamide mononucleotide. And then there's one more step with NM and NATs, convert that one to NAD. So nicotinamide mononucleotide typically had been considered just this intracellular precursor or intracellular, intermediate in the synthesis of NAD. It's now one of the more popular supplements because it's been sort of recognized that if you take forms that have the ribose already attached to the nicotinamide, it's less energetically costly to get those converted into NAD, and you can bypass the first enzymatic step that is pretty energetically costly through NAMPT. So the debate in the field has been whether nicotinamide mononucleotide goes directly into cells intact or whether it gets dephosphorylated to nicotinamide riboside and then taken up. And then those two supplements sort of compete based on this idea that one of them is the more proximal precursor because you either take up nicotinamide mononucleotide directly or you convert it to NR, in which case NR is the more direct precursor.
I'm glad you're having trouble saying these words because so am I. I'm glad also that you broached the subject of supplementation because we can supplement with NMN, as you said there. Can we also supplement with NAD?
Yeah, that has been done as well. That's not been as well studied in well-controlled experiments in laboratories. There's been a lot of small anecdotal trials in many cases with no control group and more like case reports. But people certainly have supplemented NAD directly, and there's even people taking intravenous NAD. Which has become sort of a fashionable thing at designer clinics. But again, no published data at all on the results of those studies, no controlled studies. So it almost certainly does boost your NAD levels, but it's whether it just breaks down to some of the simpler precursors or actually has any different effects because it was intact is really unclear right now.
I suppose this whole area is open to exploitation from companies looking to take advantage of the lack of concrete science when it comes to supplementation with things like NMN and NAD.
Yeah, I mean, I would say particularly with NAD. I mean, the all of it is lagging behind a little bit. The clinical trials are small and slow, but for both nicotinamide mononucleotide and the riboside, there are clinical trials in progress. We're building up a big enough body of work to sort of say something intelligent based on actual data most of the time for those two. But when it comes to comparing to NAD, I think that there's still extremely sparse evidence for anything with NAD directly.
I did read that NMN could mitigate against the oxidative stress and DNA damage and inflammatory responses. Would you concur with that?
Yeah, I mean, well, for sure in rodents. I mean, in simpler models. There is some evidence for anti-inflammatory activity in humans, but again, it's often things like treating and then isolating, PBMCs, so the white blood cells, and then stimulating them in a dish and showing that they're less likely to make inflammatory cytokines. So there's certainly evidence that's leaning that way that they might be useful as anti-inflammatory agents, but it's pretty weak as far as the human side so far. We need bigger studies and more ones that are really designed to answer that question instead of cases where people have taken advantage of a different study to look at some anti-inflammatory phenotype.
I did find one study online from the tail end of 2022 that chronic NMN supplementation elevates blood NAD levels and alters muscle function in healthy older men. Would you be familiar with this study?
I'm not sure if I know the specific one you mean, but there's multiple showing for sure that you elevate NAD levels in the blood with NMN or nicotinamide riboside supplementation. So I think that's pretty clear at this point. The blood cells will get higher NAD levels from those treatments. The improvements in muscle function in general, across a few different studies at this point, have been pretty weak. I mean, they're encouraging enough, I think, to do larger studies, but I think that's as far as I would go from from what's already published. In most cases, it's, often where they do 20 measures of muscle function and one of them is nominally significant, right? Which means a p-value that implies that it was at least a 1 in 20 chance, getting data that unlikely. So, I mean, the multiple comparison corrections haven't always been done very well.
Another term that crops up whenever I do research on aging is the term Sirtuins. Could you tell me what this is and, again, its significance?
Yeah, so this is a class of enzymes that use NAD as a substrate. So the main role of NAD that I was describing was sort of accepting these high energy electrons as you're extracting energy from food or doing other types of reactions. There's a whole separate class of enzymatic reactions that NAD takes place in, and Sirtuins are one of these enzymes that actually break down the backbone. So they cleave the nicotinamide off of it. Instead of just giving it some electrons, where it's sort of acting like a shuttle, these enzymes, like Sirtuins, break the NAD apart. So you need to resynthesize it afterwards, or else you risk depleting the NAD pool. And so what Sirtuins do with the NAD is remove other groups from proteins. So they're essentially molecular switches. So if there's something like an acetyl group on a protein, a Sirtuin can come along and use NAD as the acceptor to take the acetyl group off the protein and put it on the remaining piece of NAD, which is ADP ribose, and kick that off. So it's essentially something similar to phosphorylation conceptually. You can run around and essentially throw switches by removing these little bits of signaling molecules from proteins all over the cell, and it's using NAD as an acceptor to do that.
As I said at the outset, you're working on NAD, which is one small part of the cell, for want of a better word, and this is one key element we think and you hope is that is part of the aging process. I'm curious because there are so many different groups across the world working on aging, and they're working in different areas to you. Is there much crossover, and is there much communication between your research groups and other research groups across the world who may not necessarily be doing exactly the research that you're doing, but are working closely enough for you to be able to communicate and share information and research results?
Yeah, I mean, I think COVID made that more difficult for a while. A lot of the international meetings that have been going on for a long time got suspended, but I think it's really picking back up again now. There certainly are meetings with the theme of aging research that bring in people working on all sorts of areas. So people on NAD, but things like senolytics or calorie restriction, and we do get quite a bit of overlap between different groups there, and they're able to discuss things like what are the best models or markers for aging in rodents and things like that sort of apply to everyone.
And I suppose another important question would be, and no doubt the audience are probably thinking this, do the wheels of anti-aging research move slowly still in 2024 with the benefit of computers and supercomputers and AI and all of those things?
I think they do. I mean, if you pay attention to, especially looking on Twitter these days, right, you see one of the big fights in the field is over biomarkers of aging, right? People have wanted to take these things called methylation clocks, where you look at the methylation on your DNA and you can sort of predict the chronological age of a person based on how much their DNA is methylated. You can do different interventions and you can see if that changes. And so one of the dreams for the field is to be able to prove that is a valid biomarker, that if you do something like calorie restriction and then you measure the methylation age on the DNA of the animal like a few months later, that will tell you whether or not it was going to live longer without actually doing the whole longevity study. And so we're not there yet in terms of proving that will be true. And because we aren't, that's why the wheels, I think, in this field move so slow, because nobody's really sure anything is valid until you've done a full longevity study in a mammal, which is usually a mouse, which means 4 years before you answer an iterative question. And then you want to tweak something and do another 4 years. So there's I think a lot of hope for this methylation clock idea, but I think people have jumped ahead in many cases and started calling this biological age and saying that aging has been reversed based on changing the clock. And the field's not ready to accept that yet, and I think appropriately, we're not there yet. We don't know.
It's a really fascinating subject, and as I said, something that we all have a vested interest in. Thank you for outlining some of the basic points of aging research that have consumed you now for years, and congratulations on discovering that transporter, the NAD transporter, into the mitochondria. That's no mean feat whatsoever. But for the moment, Professor Joseph Baur, thank you so much for joining me on the podcast today.
All right. Thanks for having me. It was my pleasure.
Well, thank you for tuning into this episode of the Happy Habit Podcast. If you enjoyed it or any of the previous episodes, well, then show your support. You can like, subscribe, share with other people who you think might get value from these episodes. You can also become a subscriber over on YouTube. And if you have some free time, well, then check out some of the other videos in the Happy Habit archive. Until next time, stay happy.